This is Chapter 9, Muscles and Muscle Tissue, Part 3 of 3. In Part 1, we reviewed muscle tissue. We talked about the characteristics and the functions of all types of muscle tissue. From there, we focused on skeletal muscle. We looked at the gross anatomy as well as the microanatomy. In Part 2, we studied the sliding filament theory or model. We introduced nerves briefly. We looked at the parts of the neuromuscular junction as well as the events that occurred there, and finished up with the action potential along the sarcolemma. In Part 3, we will tie everything together by shifting our focus from the excitation part to the contraction part of excitation-contraction coupling. We briefly mentioned in Chapter 9, Part 2, the definition of excitation-contraction coupling, and we began to get into the excitation events. But ultimately, what is happening here is that we have a coupling of these events involving the transmission of an action potential or an electrical current along the sarcolemma of a muscle fiber. From those events, we eventually see the sliding filament theory, or muscle contraction. We left off in Part 2 with the action potential, the generation of it, as well as the propagation. So picking up where we left off there, now we are going to begin to see the contraction events, starting with this propagation of the action potential. As this electrical current is spread along the sarcolemma of a muscle fiber, like the frosting on a cake, that electrical current is going to dive down into the T-tubules. Remember the T-tubules? Once that action potential dives down into those T-tubules, it communicates with the terminal cisterns of the sarcoplasmic reticulum. The action potential is going to stimulate the release of calcium from the sarcoplasmic reticulum. That calcium will be extremely important. I'm now going to use this slide to review everything that we've talked about so far with regards to the neuromuscular junction and action potential, as well as excitation-contraction coupling. So take your time to follow along with me on this slide. Looking at the structure in yellow, we have a nerve, or a neuron. A neuron is made up of many axons. So what we are looking at here is one single axon in yellow that will branch once it reaches the sarcolemma, or once it reaches a muscle fiber. The one bouton that you are seeing is a single axon terminal filled with synaptic vesicles. Within these synaptic vesicles, we find a very important neurotransmitter known as acetylcholine. Once that nerve is stimulated, we will see the release of acetylcholine from the axon terminal into the synaptic cleft. Acetylcholine will diffuse across the synaptic cleft and bind to acetylcholine receptors on the sarcolemma. Here we have our end plate potential. Once acetylcholine binds, we see the opening of sodium channels. Sodium flows into the muscle fiber, driving its value towards threshold. Once we reach threshold, we are able to continue to generate and spread out that action potential. Now follow the pink arrow. The pink arrow shows you the propagation of the action potential, or the nerve stimulus. The action potential will propagate along the sarcolemma and dive deep into the T-tubules. As the action potential dives deep into the T-tubules, it stimulates the release of calcium from the terminal cistern of the sarcoplasmic reticulum. Earlier, I mentioned that calcium was a key component in muscle contraction. But without calcium, we have, remember that regulatory protein tropomyosin? Tropomyosin is blocking the myosin binding sites on the actin filament. So without calcium present, those binding sites are covered. With no calcium or low calcium present, those myosin heads cannot attach to the actin, forming a crossbridge. And also at low calcium levels, the muscle fiber is relaxed. It is not contracting. Remember, calcium is stored in the sarcoplasmic reticulum of muscle fibers. In response to an action potential, the sarcoplasmic reticulum will release calcium, and we will see in just a moment what that calcium does. Once calcium is released from the terminal cisterns of the sarcoplasmic reticulum in response to an action potential, calcium will bind to troponin. Once calcium binds to troponin, troponin will change shape or reconfigure itself. And when it does, it actually pulls the tropomyosin away from the myosin binding sites on the actin filament. Once those binding sites are exposed, the myosin heads will swing up or swing down to attach itself to the actin filament. Once that happens, we have formed a crossbridge. Our thin and our thick filaments are attached to each other. Once we have formed a crossbridge, we will now see a crossbridge cycle, which is ultimately muscle contraction. Here is where we will see the sliding filament model and the shortening of a muscle to generate force. Once we no longer have a nerve stimulation, once we no longer have calcium, we don't see muscle contraction anymore. Picking up where we left off, as that action potential propagates along the entire sarcolemma and dives deep into the T-tubules, it stimulates the release of calcium from the sarcoplasmic reticulum. Once calcium is released from the sarcoplasmic reticulum, it will bind to troponin. Once calcium binds to troponin, troponin will reconfigure itself. And when it does, it pulls tropomyosin off of the myosin binding sites on the actin filament. Once those binding sites are exposed, we are able to form a crossbridge, meaning that myosin head can now link up with the actin filament. At the microscopic level now, muscle contraction can be seen in these 4 steps, known as the Crossbridge Cycle. In order to initiate the Crossbridge Cycle, we have to first form a crossbridge, and this occurs after calcium binds to troponin and after tropomyosin is pulled off of the binding site. Once that myosin head links up to the actin filament, we have formed a crossbridge. From there, we will see the myosin head pivot or change direction. And when it does that, it pulls the actin filament towards the M line of the A band. This is known as the power stroke. Once that myosin head has pulled the actin, it can't do anything else with it, so it detaches itself. And in order to detach the myosin from the actin, that requires energy, also known as ATP. From there, the myosin head resets itself into this high energy state, and it prepares itself to form another crossbridge with a different region of that actin filament. The very last part of this lecture and the very last part of chapter 9, we have a diagram of the crossbridge cycle. At the top, we have step 1, the formation of the crossbridge. Again, calcium is released from the sarcoplasmic reticulum. Calcium binds to troponin. Troponin changes shape and by way, it pulls tropomyosin off of the binding sites. We are now able to link up our myosin head to the actin filament. From here, we have the power stroke. The myosin head pivots, and when it does that, it pulls the actin filament towards the M line. Afterwards, we have the detachment of the crossbridge, which requires energy, or ATP. From here, the myosin head resets itself, or cocks itself back into a high energy state and prepares itself to form another crossbridge with a different region of that thin filament. This was Chapter 9, Part 3. It was awfully short, but we covered the remainder of excitation contraction coupling, and we focused on the crossbridge cycle.